All right, so with gel electrophoresis, an essential idea to understand is essentially how we can use these enzymes called restriction enzymes. So a restriction enzyme is an enzyme that can cut DNA. So the idea of this would be, this is showing how we could cut a bacterial plasmid. This restriction enzyme recognizes a specific DNA sequence. In this case, this restriction enzyme recognizes AATT. So that restriction enzyme, anywhere along a chromosome, whether it's a circular chromosome like a bacterial or a linear chromosome like ours, it'll recognize this AATT sequence. Different restriction enzymes represent different sequences, and it'll then be able to cut the DNA. And so it's going to cut the DNA here, and it's going to cut it here. Now, how it cuts the DNA, you end up with these things called sticky ends, where the idea of these sticky ends is this creates almost like an anchoring point where you could insert DNA there. When we go over bacterial transformation, I'm going to go more into like how we basically could use this to insert genes and use it for genetic engineering. How this relates to gel electrophoresis is just this idea that we can use restriction enzymes as they're like molecular scissors, where we can use them to cut DNA. And these restriction enzymes, let's say you use the restriction enzyme that cuts DNA, AATT. If you do that using my DNA or say your DNA, say it's a crime scene, and we have the sample of DNA from the crime, and then we have 4 other DNA samples, this restriction enzyme will cut at different points along each suspect's DNA compared to the sample DNA from the crime scene. So, with that in mind, next slide. So here, just kind of real quick, this is showing what I was saying with like how we can insert genes. So I'm not going to dwell on this too long because we're going to talk more about this when we do recombinant DNA with doing bacterial transformation. The idea of those sticky ends is then you can then insert a gene that you also cut using the same restriction enzyme. So then they will have a sticky end that's complementary to the sticky end over here of the DNA you originally cut, and so then that allows them to be glued together. This isn't really that relevant for gel electrophoresis, but just kind of the idea of the sticky ends. But I'll remind you of that when we do bacterial transformation. So we get these fragments. These fragments are what are key for gel electrophoresis. So, then ligase will glue together if you're going to insert a new DNA strand. Anyways, gel electrophoresis. So how this works is you've got your DNA, and the DNA is of different lengths. It's of different lengths because we use that restriction enzyme, and we said that restriction enzyme recognized, I think, AATT. Could be any other sequence. So where the AA and TTs are in the sample DNA from the crime scene and each of the suspects' DNA is going to vary, which means you're going to have different lengths of fragments. Okay? So the idea of gel electrophoresis is we're going to use a gel and we're going to separate out that DNA based on the size of the fragments. Because let's say I have a DNA sequence and there's an AATT, I make a cut. Until we get the next sequence of AATT, let's say for me it's right there. For you, it might be right here. So I would have a fragment that'd be this big. Your fragment would be that big, is the idea. That's why the fragments will be different sizes. So the idea of gel electrophoresis is to separate out the DNA fragments by size. And so what you'll end up getting, and I'll come back to explain some more of this, is these different banding patterns based on the size of the fragments that we cut with the restriction enzyme. So if this was, say this was the crime scene DNA right here, these are the suspect DNAs, you could then see, okay, this, this sequence right here, or this one right here lines up best with that sequence right there. So that would be your suspect, or that would be the person who did the crime. So you can match it up with whatever your sample DNA you're trying to compare it to. Okay. So going back to how exactly the gel works and separates it, you need to remember that DNA has what charge on it? Think of the sugar phosphate backbone. So phosphate Anybody know what charge phosphate would have? Virginia?
Oh, I was just going to say the DNA is negative.
Yeah, it's negative because phosphate, that sugar phosphate backbone, has a negative charge. So we take advantage of the charged nature of DNA by running this electrical current. So we start DNA at the negative end. The negative end here is the cathode. Okay? It seems it's kind of backwards because you would think like cation's positive. Here the cathode's negative. So it's kind of backwards what you might think. Anode here is positive. So think of it as the backwards of cation, anion. Anyways, we start at the negative, and then when you run that electric current, you then will get a positive end of the gel. So opposites attract, so that negatively charged DNA is going to be attracted to the positive terminal of the gel. And so as that DNA is moving, smaller fragments of DNA, let me explain actually go into this. Smaller fragments, like this is the negative end, this is the positive end. Smaller fragments of DNA travel further, so these would be smaller. These would be larger fragments of DNA. Because the bigger you are, if you're a smaller DNA, this is a gel, smaller fragments can weave their way through the gel easier, so they can make it farther along the gel for the time period that you're running that current, compared to larger segments of DNA that are bigger, and so they have a harder time navigating through the gel, so they don't make it as far. Wells, these are where you insert the DNA. So basically wells are these like these holes in the gel where you're then going to, you'll have a pipette and you'll insert your DNA sample, and this well, however many samples of DNA you have. Usually there's like our gels, there's 8 wells. So you could run, you could run 8 different samples of DNA. But I suppose you could have bigger gels where you could test multiple, like more than one different sample, more than 8. Anyways, now what they're looking at under here is a UV light. So what you'll end up doing is you're going to dye these gels, and the dye of the gels will help you see the DNA. So you'll mix the DNA. So this DNA is mixed with dye. Mixed with dye. And that dye will fluoresce under UV light. So that UV light will fluoresce the dye, and so then you can see those bands of DNA. Okay? And again, the purpose of this is, or an application of this would be in like crime scenes and stuff, where they could then compare the sample of DNA at a crime scene with, with like the different suspects' DNA. Any questions so far? Let me make sure I said everything here. There's a buffer solution that this gel floats in. Not super important. It's an agarose gel. Not particularly important, but make sure you understand that the direction DNA moves and that it takes advantage of the different sizes of DNA. Understand smaller travel further, larger travel not as far. Okay, that's everything for this. So let me stop the recording here.